You who are here in the auditorium today and those who are joining us by live webcasting. And as you all know, asthma is a highly variable disease. And we differ in terms of our risk of developing asthma. Persons with asthma differ in their clinical presentation, the disease severity, response to medications. And what we're hoping to do today is to explore the way in which racial variation can help us to understand the genetic basis for some of these differences. And we have an absolutely outstanding panel of experts to discuss this topic, and I'd like to introduce them to you in the order that they will be speaking. We'll have 3 brief presentations and then a panel discussion. The order in which they'll be speaking happens to be the inverse in terms of the distance traveled to be here. And so first is Dr. Benjamin Raby, who is a member of our Partners Asthma Center. He's part of the Brigham division, the Channing division of network medicine. He's recognized as an expert in asthma genetics, and he's also a wonderful teacher of genetics to the non-geneticist. Second, Dr. Michael Wechsler, formerly of Partners Asthma Center, now head of the asthma section at National Jewish Health in Denver. He's been part of the Asthma Clinical Research Network for many years, and on the side happens to be world expert in Churg-Strauss syndrome. And finally, a warm welcome back to the Brigham to Dr. Esteban Burchard, who comes to us from... we did the lukewarm welcome, now a warm welcome. I'll give you the hugs later, just hang on there. So Dr. Burchard, who was a Brigham house officer on the tutelage of Dr. Marshall Wolf, went and worked in the laboratory of Dr. Jeff Drazen, and then returned to UCSF, where he is professor in both the schools of medicine and pharmacy. He heads the UCSF Pulmonary Genetics Laboratory, and he is head of their Center for Genes, Environment, and Health. And I'm very delighted that you've come back to join us. I have gifts that I wanted to share with you, our speakers. Partners Asthma Center scarves. Mike, I see you have a scarf. For you, a tie.
Thank you. Thank you very much. Wow. What do you do with the tie?
So we begin our program then, please, with Dr. Raby talking about asthma, genetics, and race.
Thank you very much, Chris. So because there's 3 of us, and I think we all have a lot of information, we're going to try and jump to this right away, and I really appreciate the invitation. We thought about doing this, I guess, almost a year ago, and I think it's a very exciting topic. So the motivation for having this discussion is clear. There are obvious differences in the prevalence of asthma depending on racial identification. And in addition to seeing variation in the prevalence between blacks and whites, even within Latino populations or Hispanic populations, one sees wide variability in the prevalence. In addition, and probably equally if not much more importantly, is substantial racial differences in both the morbidity and the mortality. And these rates are really quite frightening, the differences here. And so it's very important for us to really understand why these differences exist, both in terms of understanding the etiology of the disease, its natural history, but also how to better help patients. So whereas the 2 following speakers will be talking about specific implications related to asthma in the clinical and research setting, what I'd like to do is first give you an introduction to the concept of race in the context of genetics, a hotly debated topic and controversial topic, and then talk a little bit about, specifically with relationship to asthma, how we can think about these issues. So according to the most important reference that we have available, Wikipedia, we can talk about race in terms of a classification system that is formally used to categorize individuals or groups of individuals into discrete subgroups. And these classifications have typically been made on the basis of characteristics, anatomical characteristics that we can appreciate visually, as well as genetics. But in addition to that, race can be defined in terms of subdivisions by geography, by culture, social behavior, and even linguistic patterns, such that two tribes, for example, that don't live too far apart from each other, that phenotypically look similar to one another, may have very different linguistic patterns, and as a result, mating opportunities are less, resulting in genetic diversity between the populations. So the interest in race has been there for many centuries, in fact. The first racial categorization formally was done in the 17th century. But the interest in race was accelerated and motivated primarily by Darwin's theories of evolution, and soon thereafter, at the beginning of the 20th century, by Gayrod's description of Mendelian patterns of inheritance of metabolic traits and disease. And we all know that there are clear segregations of certain diseases on the basis of race, such that it would be very unusual to see sickle cell anemia in someone of Caucasian ancestry, and similarly, Tay-Sachs disease is typically thought of as a Jewish disease. But the question now remains whether or not these same types of patterns also play out in more complex traits. And the motivation over the past 100 years has really been to understand what the relationship is between race, genetics, and disease. And whereas it may not be politically incorrect or controversial to describe whether or not there might be differences on race on the basis of sickle cell anemia or asthma, certainly when it comes to things like intellectual aptitudes and other abilities, there's quite a lot of controversy. And of course, all of this is in the shadow of a terrible history over the past 100 years, that of eugenics. The idea that we can study population genetics, understand the population or genetic basis for bad behavior, immoral behavior, or what have you, to try and make a better society. And this was taken very seriously at the turn of the century. Francis Galton, a Harvard University professor by the name of Charles Davenport, who was in fact a member of the National Academy of Sciences, argued for the sterilization of individuals who were thought of as being unfit or undesirable, leading to the sterilization, in fact, of more than 60,000 Americans. And of course, much of this evolved into the thinking of eugenics in terms of the final solution and the elimination of gypsies, homosexuals, and of course the genetic communists. And we can see that all three of these people were scientists, so-called scientists, and in particular thought of themselves as physicians. And so when we have these conversations and we do our studies, we have an enormous obligation to make sure that we are very clear in what we say so that we don't run into these problems in the future. So what I'd like to do is spend a few minutes simply talking about how a geneticist perceives population differences. So here we have different people around the world. And the first thing that a geneticist or an anthropologist would do would look at phenotype. And on broad, rough categorization, we could come up with people in this part of the world look differently from people in this part of the world who look differently from people in this part of the world. And so we would call these races. But if we look more closely within each of these races, we see that because of deserts, or because of water, or because of mountain ranges, we have separations and subdivisions where mating opportunities between these groups is less than the mating opportunities within. And so we start to see even greater similarity within each of these subgroups, these within-race subpopulations. And so the question is how a geneticist perceives phenotypic variability across the population in the context of the genetic variation and diversity across the population. So if we think about the total genetic variability as the variability of the species, we can consider three sources of this variation. The most broad one is that of between the races. And the real question that we have here and what we're going to try and answer here is how much does race contribute to the total genetic diversity of humankind? So that would be called the sigma of race. Then there's the diversity within races, but between populations. So this would be the between population variability. And then, of course, there's the variability between any two given individuals. If we randomly take two people who are within the same population, the same subgroup, how much variation in their genome is there? And so we call that the individuals between individual variation. And so total variation is the sum of these three. Until long after the Holocaust, one was not able to really separate these three out. But in 1972, Richard Lewontin did this by looking at the different flavors, the different alleles of 17 proteins, most of them blood proteins, ABO blood group, Duffy, and so on. And he was able to partition out these three types of variation. Where does the most of it, the bulk of it, exist? Well, in fact, it's not between the races. It's in fact between individuals. One would think, well, there's still 15% left. Of that 15% left, 8% of it is still within race between the populations. And only a very small amount, 6.3%, is between the races. And so the overwhelming majority of the genetic diversity across the world is not racial in origin. Okay? This is a very important point. And what this plays out, if you look at any common allele, the vast majority of common alleles are seen across all populations. The rare ones aren't, and we'll get to that in a moment. But here's an example of the beta-adrenergic receptor. Its allele frequency is different across different populations, but it's not so different that if you had the genotype of an individual, you could tell me what their race is. Or that if you had the race, that you could tell me what the most likely genotype is. Because you would be off more likely than you'd be on, unless you just always talked about the most common variant. So this is a very important concept. And it's a powerful message that has great appeal from a social point of view, that we're all more the same than we are different. But for many people, this does not ring true. Why? It's obvious. Because we don't look the same, and we know that some of those traits are based on genetic differences. Moreover, like I mentioned before, there are mutations that differ in their frequency between the races, and we think about these in an important way. More formally, if we look at a survey of 300 genes that were deeply sequenced in hundreds of individuals, and all of the mutation characterized across Africans, Asians, whites, and Latinos, one sees that the blue color represents variants that are only seen in that one population. We see that for the bulk of variants in Africans, they're only seen in the Africans. And a substantial proportion in each of the other ethnic groups is only seen in that ethnic group. So what if a large proportion of phenotypic variation is actually due to those variants? Then genetics plays a really important role. We don't know the answer to this. What's even more frightening is I talked about what's between and within subject variability. Well, if we do the most closest relative to us that's not human, the chimpanzee, the variability between the races and the humankind and chimpanzee is 18% versus 15% for between the races. So this is not something that we're all very comfortable with, but is a fact of life. Finally, a really important insight that was first observed about 15, 20 years after Lewontin's findings was that while the individual polymorphism cannot be used to predict differences cannot be used to categorize people properly, if you take common variation and use the combinations of those variants, and this is through an analysis called principal components, you can very accurately predict somebody's racial ancestry or genetic ancestry based on common variation. Okay, so what this tells us is that the common variants in of themselves are not singularly predictive, but many common variations together make a difference, and rare variants certainly make a difference. Highlighting this again, just to show you the scale here. So these are plots of, three genetically ancestral groups, Africans, Asians, and Europeans. And then within the European group, there's an orange signal of additional people that were part of a large case control study. Here, you can see that the sigma of race, based on all of this data, is very high, right? And would suggest that racial variation is really important. But there's an issue of perspective here, because if we zoom in on this part of the figure, we see that there's wide-scale diversity even within the Caucasian group. And in fact, if we plot this on slightly different axes, this is just natural data, how it comes out, we can actually see that among Caucasians, we can generate a map of Europe, a very nice one. And so here's a very important point, that what really drives your genetic diversity and what makes you different from the next population is how likely you are to mate with someone in that population. Okay? And so the closer you are together, the more similar you will be. And so much of the biological basis of race would dissolve if everybody just mated with each other. This also plays out in expression. So this is now a principal component analysis, very similar to what I just showed you for the genetics, but at the level of expression, which are phenotypic traits, how much of a gene is turned on or off. And we see that along this major principal component, there's a division between our white and our African subjects in our study. And in fact, when you look at the genes that are differentially expressed between the whites and the blacks in this study, we see very important pathways relevant to asthma, including innate immunity. Moreover, if we don't adjust for the fact that the racial differences, in this case in a study of lung function as a function of expression, we would have many false positives that upon adjustment for race and other covariates just falls away. So there's clearly something going on here. The bottom line, and I only have 2 more points to make after this, is that the vast majority of common variants are shared across populations, independent of race. Combinations of variants and the majority of rare variants segregate between populations. And these later differences do explain a proportion of phenotypic variability between populations. The real question is, and what the rest of this conference is about today, this seminar, is what does asthma follow? Is it a monogenic disease like sickle cell? We know the answer to that is not. Is it caused by combinations of alleles? And how does race fit into this? So the only insights that I'm going to provide on this are a summary slide of the meta-analyses of GWAS studies that have been done. These are the top hits of asthma susceptibility genes that have been defined by the most powerful asthma studies done to date, asthma genetic studies done to date. And what you see is for each gene, you have the odds ratio, the risk conferred for asthma, the confidence interval, and then the minor allele frequency in each of the populations. And there are many take-home messages here. The first is, unlike cystic fibrosis, asthma is caused in part by genes with much higher allele frequencies that are common across the populations. And the effect size is modest. Instead of an odds ratio of 32 from cystic fibrosis, it's 0.32 increase, or 31% increase for some of these variants. Very importantly, the effect sizes are similar across the races for these variants. And so what that suggests, at least for these variants, is it's not a question of the combination of them, but rather that each one of them portends the same type of risk, okay? But there are some notable exceptions, and we have to be aware of them. For example, this variant here, which confers 34% increased risk in blacks, that variant is not present in Caucasian populations. And really, in Latino populations, it's the Latino populations where there's an important African ancestry, shared ancestry. Finally, we can see that in general, there seems to be some difference in the allele frequencies of some of these variants in blacks compared to some of the other populations, suggesting that at least for these common variants, there might be an increased burden there. The major problem we're having in asthma genetics today is that, in addition to all the other disparities, is the fact that we have an ethnic disparity in terms of the number of studies that have been done. Many of the studies that have been done, the majority of studies that have been done in whites. We need far more done in non-white populations to really get at this problem completely. And the last thing I'll point out is that genetics are important when we talk about race, but they're not everything. Here's the prevalence of smoking in the US population, which shows just as great, if not greater, segregation in terms of frequency among different ethnic groups than do some of the genes we've been talking about. And that is also true for many of the other risk factors related to asthma. So when we talk about the genetics of asthma and we then talk about the effects of race in asthma, we have to keep these things very clear, very distinct, very separate, and start thinking about how these exposures interact with the genes that we're studying. And the last point, this is a similar plot to the one I showed you earlier, but in a study of 1,500 asthmatics taking part in our Asthma Bridge study. And in addition to the clear segregations of African Americans from Europeans, from Asians, there are these, and the Hispanics, there are these pink dots. Those are the people who, when they reported on their questionnaire who they were in terms of their race, they said they were mixed. And what we're seeing in North America is more and more people who are actually of mixed ancestry. And so it's going to become, over time, more of a moot point from a clinical standpoint. And what we really need to start doing is actually directly characterizing the variants. And instead of classifying people on the basis of their skin color when it comes to making clinical decisions, do it on the basis of their risk factors, what they're exposed to in their life, and the specific genetic variants that they have. And so with that, I'll stop and I'll take questions during the panel later. Thank you.
So, people often remember only the first 15 minutes of a talk and the last 15 minutes of talk. So my job, I guess, is to try to make you remember a little bit the middle 15 minutes of this talk. It's great to be back here. It's nice to see all sorts of so many familiar faces and to be with such esteemed colleagues. Just in terms of disclosures, I have received all the Partners Asthma Center paraphernalia. Chris gave me the tie. I've gotten the mug, the hat, the umbrella, the frisbee. I've been fortunate to receive funding from other sources. I hope to continue to receive some funding from a variety of different research sources as well to continue to our research project. This is the view from my office in Denver, however, which is a little bit of a nicer view. I'm going to talk about a little bit about heterogeneity of response and about asthma, and particularly asthma in blacks. People are fairly familiar with this distribution of response. There's a lot of heterogeneity, and there are good responders and bad responders to different drugs. This is a study from 13 years ago. Kalin did a study 8 years ago, also again showing that there are some responders and some non-responders to inhaled steroids, or responders, non-responders to Montelukast. When you take a population, you see that this is a typical study population. It's a mixed group. And the results of all of those studies are based on population of blacks, of whites. And I've always felt that it doesn't necessarily make sense, that there's a lot of variability within populations, and we shouldn't necessarily say that everyone is going to respond the same way. And there are examples in other fields where, in cardiology and whatnot, where there are differential responders. And if you look at the asthma guidelines, they've been developed for entire populations without any consideration for any differential response by race. The studies that have been done, the pivotal studies done by the drug companies, by the Asthma Clinical Research Network, by the CARE Network, by all the different networks, they've basically taken all comers and lumped everyone together. And then the guidelines have then gone out to major populations, said, okay, this is how we're going to treat everybody, without any recognition that race may be a contributor. However, as Benji pointed out, blacks do bear a disproportionate burden of the morbidity of asthma as compared to Caucasians. They experience more urgent care visits. They have higher rates of hospitalizations. They have higher death rates. Their asthma attack prevalence of blacks is much higher than whites. The hospitalization rates for asthma is almost 2.5 times greater in blacks than in whites. And the death rate in blacks is 165% higher than it is in Caucasians. There were some studies. The SMART study showed that there's increased death and serious outcomes in blacks. This is from the SMART study. The mortality rate in blacks was 4 times higher than it was in Caucasians in that study. People say, well, maybe environment is a factor, and Benji alluded to that. But while some of the increased burden of asthma may be related to differences in socioeconomic status and or access to healthcare, studies from here, from Diane Gold of the Channing from BI, suggested that this disparity in morbidity exceeds disparities in prevalence. And then in another cross-sectional study that was done in asthma in an integrated middle-class population, asthma prevalence was twice as high in blacks as compared to Caucasians, suggesting that these biologic factors might be playing a role in these disparities. Furthermore, evidence from the ACRN, from the Asthma Clinical Research Network from CARE, suggests that blacks and whites respond differently to different therapies. So not only is the attack prevalence different and blacks do worse than whites, there's also differences in terms of the differential response between blacks and whites. And that is despite equal access to care. In those studies, and all the studies that we did, blacks and whites both had equal access to care. They both had similar adherence rates, about 89, 90% adherence. This is a study that we published a little over a year ago now, where we demonstrated that across all ACRN studies, this is over 1,000 patients over the course of about 12 years, blacks ended up having more treatment failures, more exacerbations, more hospitalizations, things like that, than whites. When we looked at it stratified based on therapies that patients were getting, we found in trials in which patients were not getting any long-acting beta agonists, there was no differences between blacks and whites. See, the treatment failure rate is similar between blacks and whites. And then when we looked at studies in which long-acting beta agonists were given, the difference in treatment failure was significantly higher in blacks than in whites. You can see that the curve is much lower, suggesting a much higher treatment failure rate in blacks who were taking long-acting beta agonists. And this was seen even in the presence of use of inhaled corticosteroids on top of long-acting beta agonists, suggesting that there wasn't really a significant protective effect of inhaled steroids. In a pediatric study, the Badger study was published in the New England Journal 3 years ago now, almost, we found similar differences between blacks and whites. You can see here, these were patients who were poorly controlled on inhaled corticosteroids and were randomized to receive a long-acting beta agonist, an inhaled steroid, or a leukotriene receptor antagonist. If you look at the white population, you can see that the probability of best response was significantly greater when using a long-acting beta agonist compared to an inhaled corticosteroid. However, in the black population, use of inhaled steroids was as good, if not better, than addition of a long-acting beta agonist, again suggesting that there are differences in response rates between individuals of different races. The discrepancy between leukotriene receptor antagonists again highlights differences. Again, the black population didn't seem to respond or prefer the use of leukotriene receptor antagonists as much as the white population. In general, long-acting beta agonists added to inhaled steroids are very effective in mixed population. These are 3 different studies that were done over the last decade where long-acting beta agonists were added to inhaled corticosteroids. They resulted in improvements in FEV1 by 10%, improvements in peak flow, improvements in change in rescue puffs per day, improvement in symptom-free days. And so this suggests that in general, in mixed populations, again, when you just take all comers, that these therapies are very effective. When you take a look at some studies, and just in the last couple of years, people have recognized that there might be differences between blacks and whites in terms of response rates. There have been 3 studies done in black populations with asthma that have been published. And what we saw was that while there was maybe some subtle benefit of adding a long-acting beta agonist, the magnitude of the benefit was not nearly as great in the black populations as compared to the Caucasian populations. So if you look in terms of change in FEV1, you can see the effect in Caucasians was almost twice as high as the benefit in blacks. With regard to changes in peak flow, while the Caucasians had a 25 to 38 liter per minute improvement, the difference was only 10 to 18 liters per minute. Same thing with rescue puffs per day. Again, they benefited by 1.5 puffs per day compared to only by about 0.5 puff per day. And similarly with regard to symptom-free days. So you can see again that while there might be some benefit to these therapies, the benefit is not nearly as great in the black population as it is in the white population. So these data suggest that asthma therapies, similar to therapies for hypertension, heart failure, lupus, and others, produce different responses in black adults and black children than they do in corresponding Caucasian populations. And they suggest that blacks in general may respond less well to long-acting beta agonists than they do to inhaled steroids when these medications are used as stepped-up therapy. Now, there's a question about the effect of maybe pharmacogenetics. And this is really, there hasn't been a lot done here. So I won't talk about it, but I'll tell you that from one study that we did, from the large study where patients were given a long-acting beta agonist on top of inhaled steroid, we found, contrary to what was seen in the general population, there did seem to be a pharmacogenetic effect. Patients who are charged with the beta receptor did not benefit from addition of a long-acting beta agonist, whereas patients who were GLI-GLI did seem to benefit from long-acting beta agonists, both with regard to AM peak flow, PM peak flow, as well as methylcholine responsiveness. To see that when a LABA was added on to these individuals, the RGARDs, they didn't benefit at all. The GLI patients seemed to have a benefit. And that it was a very small patient population, but it was statistically significant. So I think it's important to do studies in blacks too, not just in whites. I think we need to look at different groups because they respond very differently. So I just want to highlight two quick studies that we are ongoing. We don't have any results yet, but one is the BELT study, the blacks and exacerbations on LABA versus tiotropium. This is a study that Ellie and I have been working on with Barbara Yan and Wilson Pace and others from the Practice-Based Research Network. It's funded by the AHRQ. We just finished recruitment this past month. 1,070 patients were enrolled in the study, and the research questions we're posing are: in blacks with asthma, is a regimen containing LABA added to ICS superior than adding a tiotropium? And also we're looking at the beta receptor and seeing whether or not there are differences in the blacks who are RGARD versus GLI in terms of their differential response. And we're basically giving inhaled steroids plus tiotropium versus inhaled steroids plus a LABA. We ended up with Be Practical. This is the study that the Asthma Clinical Research Network is going to put forth. It's blacks with poor asthma control taking increased corticosteroids and or LABA. And that's a study where we're taking all comers, blacks age 5 and older, adults and children, and who are poorly controlled, and we're stepping them up to different therapies. We're going to add a LABA. We're going to add to increasing dose of inhaled steroids. We're going to look at the response to different levels of inhaled steroids, and we're going to look at the effect of increased inhaled steroids on LABA. So it's a 4-arm crossover study looking at increasing inhaled steroids versus adding a LABA, and each subject will get that. We're also going to look at the degree of African ancestry and whether or not, and Esteban's going to talk a little bit about admixture, and whether or not that may affect preference for different therapies. And the reason we're doing that is because current self-reported blacks range from 97% to 29% African ancestry. And that's one of the highlights that Benji showed. And it's been postulated that as these populations become increasingly admixed, that differentiation based on self-reported measures will become increasingly imprecise. So we need to do things more on a more genetic basis. And a good example is Barack Obama, who's certainly of mixed race. So I'll just sum up. Clearly, black adults and black children respond differently to currently recommended asthma treatments than Caucasians. The current asthma guidelines may not be appropriate for black adults and black children with asthma. And genetic ancestry using ancestry informative markers may be a superior method to identify subjects who may benefit from one therapy or another. The Be Practical study that we're doing is going to study blacks. It's going to be also a large study of over 600 patients. And we're really going to try and address gaps in the asthma guidelines. It's fully powered to look at adults and children, black adults and kids. And we're also going to hopefully identify potential genetic biomarkers. So now I'd like to pass on to Professor Esteban Burchard.
Well, beautiful. It's an honor and a privilege to come back to what I consider my home turf. My scientific career in asthma and my interest in asthma had its genesis here. The Gala study started here when I worked in Jeff Trace's lab. And I had long been interested in health disparities, and I got trained in genetics at Stanford. And when I came into Jeff's lab, I was unsure of what I wanted to do. But once I got into a project in Jeff's lab, I was able to merge what I call academic rigor with personal passion. And we started the Gala study, and we've never stopped. So I want to talk about the scientific advantages of studying racially mixed populations. Now, we know that, for example, there are disparities in diabetes across populations. And so if you're doing genetic research, let me posit a question to you to think about while I'm talking. If you're looking at the insulin gene, do you think it matters if the insulin gene is of Native American ancestry, European ancestry, or African ancestry? And I'll argue that it is. So I have no financial disclosures, but I'm available for sale if anyone has money for me. When we look at the demographics of the United States, it's changing dramatically. In fact, asthma is the most common pediatric disorder in children. 47% of children in the United States are minorities. And as of today, more minority children are born every day than Caucasian children. So it's important to keep that in mind when we look at research. When we look at genetic research, in the last 5 years, we've identified more genes through genome-wide association studies than we have in the history of modern genetics. And we published this in Nature last year. And this is actually important because 96% of all genome-wide studies have been done in European populations. And not only from a social point of view, that's a social injustice, it's a scientific injustice. Particularly as a physician scientist, when you look at differences like this, in the United States, asthma prevalence is highest in Puerto Ricans, lowest in Mexican populations, although the vast majority of research focuses on these populations. Now, as an epidemiologist, you might argue, well, it's reporting bias and so forth, but it's really hard to argue when you look at death rates. Again, you see the same patterns. And Jeff and I saw very preliminary data like this back in 1998 at the ATS in Chicago, and it really inspired me. And so what we did is we created the genetics of asthma and Latino American study at the ATS, and Jeff put me in front of Claude Lafont, said you got 2 minutes to present your idea, and let alone we ended up writing the grant, and before I left the Brigham, we had funding for the GALA study. I subsequently went on to start GALA 2, and we now have an African American study. And what we did is we built a network of individuals that are minority-serving individuals across the United States, and we recruited locally, nationally, and internationally. We still have this network going. And what we've done is we created the largest minority pediatric asthma study in the United States. We have over 7,700 kids, 9,100 individuals, African Americans and Latinos. And what you're looking at right now is the executive board, and I have the ethos of trying to make this a publicly available resource. My only criteria, if you want to collaborate, is that you're nice and you're fun to be with. Okay, so you don't have to go to like Cardia and all those, you have to go through all the hoops, you just go through me. But when we talk about Hispanic populations, they're racially mixed, and Benji did a really good job of talking about that. Hispanic populations, the contemporary Hispanic population and African American populations, are an amalgam of 3 major ancestral groups: African, Native American, and European. And what's really cool is now with genome-wide association studies, in which you have, say, 1 million markers or 2.5 million markers, you could take every SNP as a single data point and add to some sort of analysis to come up with global ancestry estimates in individuals. And what this slide shows is that each bar represents an individual, and each color represents that individual's racial background. So here you have Puerto Ricans on the left. The red color represents Native American background. The blue represents African, and the opaque gray is European ancestry. And you can see between Mexicans and Puerto Ricans, there's a lot of variability. From individual to individual, there's a lot of variability. Now, this is what we call global ancestry. What's really cool now is we can now take an individual and assign ancestry at every locus in this individual's chromosome. So this is the Puerto Rican individual, and we have chromosome 1 at the top and 22 at the bottom. If you go from left to right, you see that their ancestral blocks are what we call locus-specific ancestry, and it alternates. It flips from European, shown in yellow, Native American shown in red, green shown in African, and it just alternates back and forth. And this is really cool because this is work that my group really pushed into the clinical arena. And one of the lessons that I learned from Jeff was, that's all good and well, but make it clinically relevant. And that was a challenge. And so how do we leverage genetic ancestry to scientific advantage and clinical advantage? Well, while we were thinking about how to do that, one of the things that would happen was the game changed. The NIH, the NHLBI in particular, forced everybody in the United States that had asthma genetic data to pull it into a pod and be one big happy family. But it was really cool because it became, this is the EVE Consortium. It became the largest, most ethnically diverse consortium in the United States. Lots of damn conference calls, but it was a very diverse study population with genome-wide data. And you know, what we did out of that, we did the standard Nature Genetics paper with 120 authors. And it was impressive, but not very impressive because we only found one gene that was specific to African Americans that increased risk for asthma that was not found in other populations. And Dr. Raby talked about that. But then we started scratching our heads and we said, we believe that there are biologic differences in health disparities, and we didn't see them in the EVE meta-analysis. And so one of the questions that one of my outstanding graduate students, Chris Genu, did is he said, okay, well, why don't we look at ancestry rather than genotype and regress that against the phenotype that we're interested in. So we took all the Hispanic participants in the EVE study, we took all the African Americans, that's about 7,000 individuals, and we did an admixture mapping meta-analysis. And again, Chris went back and measured local specific ancestry on everyone in the EVE Consortium, and we just regressed that against asthma. And what was really cool is we got this huge hit on chromosome 18 that met genome-wide significance. And remember, for admixture mapping, since we're doing fewer tests than traditional GWAS or allelic associations, the threshold, the Bonferroni threshold for statistical significance is much lower. So we don't have to meet the 10 to the minus 8. We can meet our empirically derived threshold. And here, this is a clinically or a statistically significant peak. Now, what we did is we looked underneath that peak and asked what were the genes underneath that peak that were driving the associations. There are two genes, and one of them, SMAD2, is in the TGF-beta pathway. And what's really neat is SMAD3 had been identified in European populations through genome-wide association studies, but there's not been a single study out there that's implicated SMAD2 being associated with asthma, at least from a genetic perspective. And so that was nice. Then what we did, what's more important than the actual gene is the ancestry at that particular gene. You can see here, these are odds ratios. You can see here that if you're African at that particular gene, you have no increased risk. If you're European at that particular gene, you have a decreased risk. If you're Native American at that gene, you have an increased risk. So go back to my idea about the insulin gene, whether it matters if you're Native American, European, or African. Well, here's a case where ancestry at that particular SMAD2 locus matters. So then that gave us the green light to proceed. And because we have one of the largest Latino cohorts in the United States in my lab, we were able to tap right into the GALA2 study population, 4,000 individuals, and replicate the association. And the association is strongly replicated, 10 to the minus 4. And it replicated in the same direction. Native American ancestry was a risk factor. European ancestry at that particular locus was protective. So that gave us the green light to take on another experiment in which we looked at gene expression in peripheral blood between cases and controls for SMAD2 gene expression. And what we saw is that amongst cases, they had a much lower level, 25% lower of SMAD2 gene expression than in healthy controls. So again, what we're finding is that ancestry matters, SMAD2 matters, and it correlates with differences in gene expression. Now, with the GALA2 and the GALA1 study, we were able to look at all the risk factors that we've identified as being significant for the development of asthma. And we wanted to look at what we call the population attributable risk. And we see that obesity over here has a very high population attributable risk. Nitric oxide, which we measured air pollution at every clinical site that we recruited at and every home residence, has a pretty significant risk. This is the canonical ORM-DL3. That's why you see a very tight confidence interval. SMAD2 is next. And this is much higher than risk of in utero smoking for the development of asthma. So it suggests that this particular locus, which is Latino specific, is an important risk factor for the development of asthma in these populations. And so to sum up my take-home point, ancestry, using a new technique, not allelic associations or traditional genome-wide associations, but here we leverage locus-specific ancestry. We're able to take a novel analytical method, apply it to existing data to identify novel risk factors for Hispanics. And what's also cool is that SMAD2 locus was not a risk factor for African Americans. And what's also cool is that when we went back to the EVE analysis, and Dara Torgerson, who was the lead author on the Nature Genetics paper, she's now in my lab, we went back and looked at the original data, and there was not even a clue that SMAD2 might be associated with asthma. So again, I want to emphasize that using a different analytical technique on existing data, that we were able to identify novel risk factors. Well, why is this important to you? You might say, well, I don't have admixed populations. Well, since 2006, the number of genome-wide association studies has increased dramatically. And what this has created is almost like a genome envy, in that there's been a genetic backlash, and I'm on study section and I see this happening. And this is too bad, because there's a lot of data and lots of people have got their paper out and are moving to the next latest and greatest toy of sequencing or exome sequencing and so forth. But what we believe is that if we can go back to those existing data and mine them using admixture mapping rather than allelic associations, that we might be able to identify untapped findings that were there. And that's what I say, don't throw the baby out with the bathwater. Well, this is my team, and I'm very grateful to have a large team, but a very, very diverse team. And what I do is I take people that are clinicians, I take people that are population geneticists, statisticians, epidemiologists, and I make them all sit in the same area. So there's cross-pollination through osmosis, and that's key. And what I really do is these are my individuals in my lab, but what I do is try to identify top talent and really let them work and get out of their way. And I'm really fortunate to be able to have a great team, and everyone works together on each other's projects to put this work together. Again, I want to reiterate, my interest in asthma started here. My career in asthma has started here, and that was in 1998. And once we started the Gala study, I've never looked back, and it started and jump-started the careers of lots of people, and many of my trainees have gone on to faculty positions. And I'm very grateful to Jeff and Scott and Ed Silverman, who helped start all this, and I'm grateful for the opportunity to come back. It's really a personal privilege and honor to be here, and thank you for listening to me.